Imagine traveling 3.5 billion years back to the dawn of life. The tiny cells you'd see then are the very same kind you'd see today. They're called prokaryotes, the living fossils that help explain how life began. In this video, you'll learn what a prokaryotic cell is, how its parts work together, and how it differs from the more complex eukaryotic cells. To begin, the term prokaryote comes from the Greek words pro meaning before and karyon meaning nucleus. In other words, these were the first cells to exist long before more complex, nucleus-containing cells evolved. Prokaryotes come in many shapes: spherical cocci, rod-shaped bacilli, curved vibrios, and spiral forms. They're usually unicellular and include bacteria, cyanobacteria, and the tiny mycoplasma. Now, let's take a tour from the outside in, starting with the surface and working toward the center. First, some prokaryotes are surrounded by a sticky protective layer called the glycocalyx. When it's thick and well-defined, it's known as a capsule, which helps bacteria avoid drying out or being engulfed. Beneath it lies the cell wall, a rigid layer made of peptidoglycan that gives the cell its shape. Under the wall is the plasma membrane, a phospholipid bilayer with embedded proteins. It controls what enters and leaves the cell and serves as the main site for energy reactions, since prokaryotes lack mitochondria. On the outside, some prokaryotes move using flagella, the whip-like structures made of flagellin. Others use pili to attach to surfaces or exchange DNA with neighboring cells. So far, we've mapped the outside: a protective glycocalyx or capsule, a peptidoglycan cell wall, a selective plasma membrane, and external tools, such as flagella for movement and pili for attachment or DNA exchange. Curious how much you're remembering? Your personalized practice test, flashcard set, and study guide are all waiting for you. Just tap the link now and keep watching. Future you will thank you. Now that we've mapped the outside, let's step inside the cell. Beneath the membrane is the cytoplasm, a jelly-like mixture mainly made of water but rich in enzymes, nutrients, and ions that support chemical reactions. Near the center is the nucleoid region. It is not a true nucleus, but a dense area containing the cell's main DNA loop. Many prokaryotes also carry small circular DNA called plasmids, which often hold genes for antibiotic resistance or other special traits. Scattered throughout are inclusion bodies, the storage granules for nutrients, and thousands of tiny ribosomes, where proteins are made. Now, let's see how prokaryotes differ from eukaryotes. First, they have no nucleus, so their DNA floats in the cytoplasm. Next, they lack membrane-bound organelles like mitochondria or the endoplasmic reticulum. They're also much smaller, often 1/10 the size of eukaryotic cells, and they multiply rapidly through simple cell division, helping them adapt to changing environments. Overall, their design is streamlined, not lacking, and simple doesn't mean inferior. These cells thrive in boiling vents, icy oceans, and even our intestines, proving that simplicity can be a powerful way to survive. To recap, prokaryotic cells are small, nucleus-free, single-celled organisms. Their key parts: capsule, cell wall, membrane, cytoplasm, nucleoid, plasmids, and ribosomes work together to keep life running at its simplest, most resilient level. Ready to quiz yourself with the content covered in this video? Check out the link in the description where you'll find Quizlet content made just for you.